be talking about sphingosine based. So again, you should be able to draw a fatty acid given the two different nomenclature ways, either with the deltas or the omegas. From there, you should be able to draw glycerol and attach 2 or 3 fatty acids and the phosphate groups either with choline, ethanolamine, or serine to make phosphoglycerides, or triacylglycerols for 3 fatty acids. Now we will talk about sphingosine. So sphingosine, by definition, is what we call a C18 amino alcohol. So sphingosine already exists with basically 1 fatty acid tail. We don't just have sphingosine out there in our systems. It's always added to another fatty acid tail and other things, okay? But if you take a look at how I've drawn the C18 amino alcohol here, or sphingosine, I still have 3 carbons on one side. I do that intentionally, so it's similar to how we do glycerol, except by definition, sphingosine is a C18 amino alcohol. So when you draw 18 carbons, you can count them with me here. 1, 2, 3, 4, 5. 5 and 12 is 17, plus 1 is 18. So you'll always have a double bond here, okay? And you'll always have 3. So you go 1, 2, 3, 4th carbon here, double bond, and then the rest. But it's a C18 amino, so on these 3 carbons, C18, you've got 1 fatty acid tail essentially here, okay? Amino on this 2nd carbon, or the middle carbon of the 3 that I've drawn. Alcohol, you have an OH group here and an OH group here. So this is why I draw these 3 carbons here, because this is very similar to glycerol, except we just replace one OH, the OH in the middle carbon, quote unquote, with an NH3 group. Okay, so carbons 1 and 3 have alcohols. Carbon 2 has the alcohol replaced with the amino group. And there's a total of 18 carbons with a double bond. So this is a mono unsaturated fatty acid if you're looking at that as a tail. Okay, so what we're going to then do is we can further derivatize sphingosine as a result. So if you take a look, you should be able to draw the structure and recognize the structure of sphingosine, either fully drawn out as I've drawn on the board here, or like in the slides with your book where they've drawn it with just the double bond with the skeletal structure. Either way is perfectly acceptable. And if you notice in sphingosine here, they draw it as NH2. You can either draw it NH2 or NH3+. Okay? So if we take a look, we now can further derivatize sphingosine. So sphingosine, remember, doesn't exist really in our membranes and things like this. We have to add particular things. These are often, again, found in membranes. So just remember, right, in your little cartoon diagram, there's always 2 fatty acid tails. Well, sphingosine already has 1 fatty acid tail. So what we're going to do is, on the amino group here, we are going to add a fatty acid tail. So now we have 2 fatty acid tails. When we add a fatty acid tail here, we make what's called a ceramide. And a ceramide, by definition, has an amide linkage, which is, again, a nitrogen, C double bond O, with whatever the rest of the R group is, to the back. So again, in order to make a ceramide from this here, you're going to add the fatty acid tail, but instead of making it an ester, you will make it an amide linkage. So you would have C to a nitrogen, H, make sure you don't forget that H there so that you have the correct structure, double bond O to the rest of whatever the R group is. So again, this would count as carbon number 1. So if you were to draw, for example, a 14-0 ceramide, you would say, okay, this is 1 carbon. I would need 13 more. Oops. So you're going to draw CH2 with 12, 13. And now we know we have no double bond, so you have 12, 13, 14 carbons. That is a C14 ceramide. So you see how we can build these pieces together. So sphingosine, you add a fatty acid on the ceramide. Now you have, again, different size fatty acid tails with that amide bond, okay? So again, an amide linkage, just a reminder, an amine with a nitrogen with a C double bond O. We're gonna see this amide linkage again when we talk about proteins. So it's very important in biochemistry looking at amide linkages. So now that we know what a ceramide looks like, so now we've got the 2 fatty acid tails. Well, now we do the same thing like we did for the phosphoglycerols. is we can further derivatize this group here to give you the head group. So we go from ceramide, but now we can, on this particular oxygen, for example, do what we did before with the phospholipids and create a phosphate. Oops. And you can here do ethanolamine, you can do choline, or you can do serine. So if you derivatize this group here, so if you take a ceramide plus a phosphate group, you make what's called a sphingomyelin. Okay? You could also take a ceramide and you can add a sugar like a glucose or a galactose or a fructose in the ring. Okay? That makes a cerebroside, which is different than a ceramide. So again, a ceramide plus a sugar is a cerebroside. A ceramide plus a phosphate group is a sphingomyelin. So you may have actually heard of some of these before. So if you've ever heard of like myelin sheaths, for example, we can sometimes talk about destruction of myelin sheaths in certain autoimmune disorders like multiple sclerosis and things like this. And so what sphingomyelins do is they can coat your nerve cells, essentially. They're part of what are called the Schwann cells, and that helps to transmit signals to your muscles, between your muscles and your nerves and things like this. And that's why some of that material, when you have things like different autoimmune diseases, we talk about inability to move muscles and things like this. If you degrade those myelin sheaths, you're not communicating between your nerves and your muscles very well. Okay, so we can have other types that we're not going to talk about in this course, but if you're interested, please let me know. You can actually take a ceramide, and if you add a really complicated sugar, not just so a cerebroside just has a simple sugar, maybe just a fructose cerebroside or a glucose cerebroside, just like one sugar. But you can make bunches of different types of sugars. You can put a polysaccharide on there, and that gives you something called a ganglioside. And a ganglioside is actually neat because we talk about ganglions or bundles of nerves. That is, again, communication between those. And so lots of different types of sugars enable you to differentiate between a bunch of different types of signals that you have. So again, if you add a simple sugar, like you can see in the example slides here, right, you get a simple sugar, for example, D-galactose. This would be a galactocerebroside or an 18-0 galactocerebroside. You should be able to draw that. So you know 18-0, that's on the ceramide. You know it's sphingosine because it's a ceramide or a cerebroside. And then you know, oh, it's a galactocerebroside. You just add a galactose on the anomeric carbon, if you remember back from chapter 14. Okay? So again, virus entry, like we talked about with sugars, right? Communication and viral entry, those happen with gangliosides because they can be attached to a lipid on the outside of a cell to get that communication back and forth. So now we have finished talking about the main types of lipid membranes. Are there many more? Yes. Are we going to talk about them? No, because it's already enough as it is, in my opinion. So if you remember, membranes, right, are known as lipid bilayers. So maybe you've heard that term bilayer before. So that's two. So we will come back next in the next video and talk about other different membrane components, such as cholesterol, the architecture of a lipid bilayer, and how this kind of goes together.